It's good stuff. Yeah, that's like dead on right. So you guys can publish that.
Yeah. I mean, it could be the good to know.
Space is like. Yeah, it's about picture as well, like that. I think I got you there, yeah? Yeah. Yes. Yeah. Well, if you, yeah, in this case, through.
I should have same my first draft.
Go. Yeah.
Okay, everyone, thank you so much for coming. I'm super excited to welcome Sarah to be our ESI State of the Art speaker this month. Thanks so much for making the time to come. This is a little bit crazy time. So Sarah's a lecturer in the Center for Ecology and Conservation. Her research focuses on plastic pollution in marine and coastal areas and its impact on marine vertebrates, such as turtles and seabirds and marine mammals. She did her PhD here at Exeter, Exeter and Plymouth in collaboration with Plymouth. And yeah, you're going to be talking to us about marine litter, microplastics, and marine vertebrates. Thank you so much.
Thank you very much, Camille. Okay, no, great start.
Do you want me to keep moving it up ahead?
Yeah, my computer's not even working either.
No problem.
Right. Yeah, okay, it's working. Hey, so yeah, random slide, but yeah, sorry. So never in my life have I actually started a talk with a squirrel meme, but here we are. So it's kind of the only way I could think of to summarize the existential crisis that I experienced when you guys invited me to give this talk, because I really struggle with kind of my identity as an academic, and it sort of got me thinking about who I am as an academic. Obviously, as Camille said, the theme of this talk series is ESI Academic of the Month, State of the Art. I'm not actually based in ESI, and I'm not sure what I do is actually state of the art. But I am affiliated to the ESI through some of my research, and I think some of the work that I do is interesting to some people. So I would kind of describe myself as a marine conservation scientist with an ecology background, who dabbles in various different disciplines, including ecotoxicology, social sciences. I also sometimes think of myself as a bit of a geographer as well. So yeah, a bit of an identity crisis, but hopefully you'll be able to see some of that kind of interdisciplinary work when I talk about my research later on. So for the purposes of this talk and my kind of research in general, I have two main kind of focuses, which are marine vertebrate ecology and plastic pollution. And during my career, these two themes have kind of either been completely separate or they've overlapped, which does make for a slightly difficult narrative. So please bear with me on that. And as Camille said, by marine vertebrates, I mean marine megafauna, so marine mammals like your turtles, your seals, whales and dolphins, and your marine turtles as well. So before going too much into my actual research, I just wanted to kind of give you a bit of a potted history of how I ended up here, because I took a bit of a convoluted route, and it kind of explains a little bit about why I am the way I am. So I started by doing my undergraduate degree at the University of Plymouth in environmental biology. At this point, I didn't really have a strategy in mind, a career plan. I was 18 years old. I just knew that I wanted to save the world. And I also knew that I liked marine turtles. So at this point, I decided to do my dissertation on the relationship between carapace size, so the shell size of female loggerhead turtles in the Mediterranean, and their clutch sizes, so the number of eggs that they laid, because I went on a turtle project during my between my second and my third year. So really liked turtles, didn't really have much of a career plan in mind. And then after my degree, I spent some time in what I like to call my lost years. So if any of you know anything about turtles, some animals go on a period of their lives called the lost years, which is normally adolescent animals go off into the world, swim out into the ocean, spend some time trying to figure out how to survive, what to do with their lives. And 2008 to 2012 were kind of my period to do that. So I spent some time traveling, volunteering, worked for different conservation organisations and environmental education charities, but nothing really felt like a long-term option. I learned a lot about myself during that time, but yeah, didn't really have a plan again. And then in 2013, I had the opportunity to come to here and study for a master's in conservation and biodiversity. And that's when things really kind of took off for me. It's when I met some really cool people, had a fantastic time and really got bitten by the research bug. So for my master's thesis, I once again chose to focus on marine turtles, looking at the potential impacts of seismic surveys from oil and gas exploration on the marine turtles globally. And for that, my master's supervisor was Professor Brendan Godley, who most of you probably know about. So he specialises in the ecology of marine turtles and other marine vertebrates. And he became and still is my mentor. And then straight after that, I got a graduate role helping Brendan in his research and education while he was head of department. And all of this kind of really fed into me and being able to get being offered a PhD at Plymouth Marine Lab and the University of Exeter, looking at plastic pollution in the marine environment and its impacts on marine vertebrates. And then after my PhD, I came back here again as a postdoc, and I think I was a postdoc for about 2.5 years. And during the first 2 years, I think I worked on maybe 4 or 5 different projects at the same time, which I think is quite an unusual experience for a postdoc. And when I say they were different, they were very different. Some of them were in biosciences, looking at the ecological impacts of plastic pollution, and some of them were in the business school, looking at more of the people side of things. So it was a really interdisciplinary experience. Obviously taught me a lot, gave me a lot of different skills, and not least just project management and how to juggle lots of different plates spinning at the same time. And then towards the end of 2021, just before I went on maternity leave, I decided to take a punt and apply for a lectureship, and amazingly got the job. And now I'm a lecturer in marine vertebrate ecology and conservation. So you may be wondering how I went from being interested in marine turtles to then dedicating my career to researching plastic pollution. In a nutshell, kind of boils down to the fact that I spent a lot of time growing up on the coast. I was always taught to put my rubbish in the bin, had a very strong awareness of recycling and things like that. But it was during my time in the lost years, when I worked on turtle nesting beaches in the Mediterranean and Costa Rica, where I really started to notice plastic pollution and how it was becoming quite a big problem for the marine environment. I did a lot of beach cleans and community engagement on the issue. And I really started to realise that this is what I felt passionately about. And then during my master's, Professor Tamara Galloway from Exeter came down and gave a talk about some of the work that her and Matt Cole and Penny Lindeque from Plymouth Marine Lab were doing, looking at microplastic ingestion by zooplankton. So that for those of you who don't know, this is a copepod. It's a type of zooplankton. They are at the base of the food chain. They're microscopic. They support the whole entire marine ecosystem. They're really important. And Matt's study showed that these animals could eat microplastics. So microplastics are defined as being less than 5 millimeters in size. Generally, they're a lot smaller than that. Sometimes they're completely invisible to the naked eye. And during the lab feeding trial, he fed these fluorescently labelled microbeads to copepods and could show that they are being ingested by the copepods and moving through the digestive system, which you can see here in this picture. And this image and Tamara's talk was something that was really pivotal to me in my awareness of plastic pollution. I had it literally blew my mind. I had no idea that plastic could be this small, that it could be eaten by animals at the base of the food chain. And it was really quite alarming for me. And since Matt's study in 2015, this scenario has been demonstrated to occur in the wild now. So we do know that this is happening outside of the lab. And from Matt's work, we know that these microplastic ingestion is known to cause issues for reproduction and survivability of these really important organisms. And then that got me to think about turtles again, always back to the turtles, and how plastic might be impacting upon them. So when I was working for Brendan, I approached Penny and Tamara and others to write this review about the potential impacts. And we demonstrated that all 7 species of marine turtles are being impacted by plastic through things like ingestion. So you've got this image here that shows all the kind of bits of plastic that were found inside the digestive tract of a juvenile turtle. This turtle is excreting some plastic, but has been shown that that large volumes of plastic in the digestive tract can impact buoyancy of these animals, which is really important for things like diving to access food and also predator avoidance. We've also got entanglement, which is this image here, and the wider ecosystem effects. You can see this nesting female is trying to dig her nest chamber in the midst of all this macroplastic, this large debris that's on the beach. Not only is this hampering her access to the sand, but it's also potentially altering the properties of the sand itself as well, and potentially causing implications for the nest environment. And I think this is all kind of fairly obvious stuff, but at the time, I think we really hit the timing just right when people were really becoming quite interested in plastic pollution. It was around the kind of Blue Planet time, Blue Planet 2 time. And interest in this paper was really, really strong. It was my first peer-reviewed publication, which was quickly followed by my master's thesis on seismic surveys in turtles. And again, this was another point at which I realised that this is something that I felt very passionately about and I wanted to pursue. And very handily, I was offered a PhD through PML and Exeter with Brendan, Penny and Tamara as my supervisors, which were an awesome team. So for this talk and kind of moving forwards, I'm going to split my plastic pollution research into 2 main themes, mapping marine litter and plastic ingestion by marine mammals. So first of all, we'll talk about marine litter. So marine litter, around 80% of which is plastic pollution, comes from a variety of different sources, including things like shipping, commercial and recreational fisheries, mismanagement of domestic and industrial waste, sewage systems, and direct littering by members of the public. Understanding where it's coming from, where it ends up, how much there is really important for feeding into kind of effective policies that are aimed at reducing it. But we need monitoring programs that provide data to highlight the kind of patterns and trends that are able to help us identify this. So beach cleans are a well-known technique for gathering information on the status of marine litter, but collecting data over large, long time periods and large geographical scales takes considerable time and resources. So to get around that, volunteers are often recruited as citizen scientists. And not only is that really effective at collecting large volumes of data, it can reduce the kind of person-hour cost of collecting those data and also removing large amounts of litter from the beach. But it also, by engaging lots of people from different backgrounds, can also help raise awareness of environmental issues like plastic pollution and potentially lead to positive changes in people's behaviours and attitudes. So during my PhD, I analysed the Marine Conservation Society's beach clean data set that involved 3,245 beach clean surveys from 736 beaches in England, Scotland and Wales over a 10-year time period. And that involved over 73,000 volunteer hours. And the map over here just shows all the little dots where the beach cleans took place. So you can see that we've got really good coverage of most of the British coast, with the exception of like some of the more sparsely populated areas of Scotland. What sets this data set apart from other citizen science projects is that we had information on the number of volunteer participants, the duration of the beach cleans and the length of the surveys as well. And that allowed me to correct the data for effort and make comparisons among all the surveys in a way that all the data were standardised. So this was really important because obviously there's a lot of different amounts of effort going into all these different surveys, and the more people you have looking for litter, potentially the more litter you're going to find, and you need to find a way of standardising that. So I won't go into any of the details about the analysis because of time, but just to kind of highlight some of the results from this, I looked at where in the British coast might we find the highest levels of, or the highest densities of marine litter. And we found that the southwest coast and the south coast of Wales had the highest concentrations. And then that was for overall litter. And then when I looked at specific litter items, I found the same pattern for fishing gear and food and drink packaging. There are probably several reasons for this, but one of them is, we stick out into the Atlantic, the Northeast Atlantic. The prevailing winds and currents are coming from the Atlantic, potentially dumping lots of debris on our coastline. But also we have high levels of fishing activity around our coastline and high levels of tourism. So lots of people using the coast, which could lead to these patterns that we see here. And then I also looked at wet wipes because Marine Conservation Society were very interested in those at the time. This was before all the kind of campaigns and stuff around people not flushing wet wipes down the toilet. As not only do they block sewage systems, they also break down into microplastics. They are made of plastic. So they wanted to know where the hotspots for wet wipes were. And we found that the east coast of the UK, this is the southern North Sea region, had the highest levels of wet wipes, potentially because there's quite a few large discharging rivers in this area. And obviously people from urban centres, a lot of people there flushing wet wipes down the toilet and then ending up in the marine environment. So yeah, that's really interesting stuff from the Marine Conservation Society's point of view and where to direct their efforts. I looked at the general changes in litter abundance over time over the 10-year time period. I didn't find anything significant for the general litter items, so all litter combined, but I did find significant increases for certain items, which were balloons, polystyrene foam, a large fishing net, wet wipes and food and drink packaging. So all of these specific items have increased significantly over that 10-year time period. And then a few years after I'd completed that project, I was approached by Natural England to repeat the analysis, but with a 25-year data set, because by that point MCS had digitalised 25 years worth of their citizen science data. And Natural England wanted me to look at beach cleans and marine litter in relation to the English Marine Protected Area Network. So this map just shows the beach cleans that took place either in an MPA or within 700 metres of an MPA boundary. And the polygons out in the sea here are just the MPAs. Don't worry about the colours and what they mean. They're just different types of MPA. So that left me with 1,836 beach cleans that took place on 646 beaches in 112 English MPAs. So just to reiterate, it's just England. So that's why you've got these gaps here for England and Scotland and Wales. And again, I was able to highlight the areas that had the highest levels of plastic litter on the coastlines. And the MPA with the most litter was the Thames Estuary and Marshes SPA, which is over here. But what really shocked me was, of the 10 MPAs with the highest plastic litter densities, 8 of them were in Devon and Cornwall, which I guess shouldn't surprise me because my previous project highlighted that this area did have the highest levels of plastic and marine litter debris. But from a policy point of view, this is really important because these areas have been designated as MPAs because they contain sensitive habitats and species. And what this shows is that they're not being protected from the potential harm caused by plastic pollution. MPA boundaries aren't physical boundaries. They can't stop plastic getting into those environments. So if we want to protect those sensitive habitats and species from plastic pollution, we need to take more of a holistic approach. MPAs aren't going to do it. So just staying on the theme of citizen science, this is a slightly different angle and it's not mapping plastic debris, but we wanted to look at how citizen science can inform our understanding of plastic pollution and what we can do to improve that. And so we held a workshop during my postdoc era and we invited representatives from 20 different organisations that deal with plastic pollution in the marine environment, and they use volunteers to collect data to some extent. And we talked about the challenges and opportunities that citizen science might offer in helping us understand plastic pollution. So from my point of view, this was the most important. What are the barriers to collecting scientifically meaningful data? Obviously, I've got experience of analysing data collected by citizen scientists. It's an amazing resource, but it's absolutely littered with flaws, and it definitely could be more robust. And so I wanted to talk to these organisations who all collected litter data to some extent. What's stopping you from collecting really robust data that you could then feed into scientific research and then onto evidence-based policies? And we talked about things like the complexity of data collection, motivation and data quality and things like that. And then we also talked to them about how we go about maximising citizen engagement. From their point of view, how can you target different demographics? So beach cleans and collecting data from beach cleans is a very white middle class activity. How do you diversify that and engage different communities? Obviously, there are a lot of people around the coastline who are really interested in this topic already. It's like preaching to the converted. How do you engage people from more inland communities, cities and things like that? So we talked about that, and we talked about whether engaging more people is even necessary, because they do have a lot of interest. But I think the most important takeaway from that was about diversity and how to improve that. And then we also talked about the opportunities. So how could these NGOs engage with different sectors like the commercial industries, policymakers, waste and recycling, to really feed into, tackling plastic pollution. And on that theme as well, we talked about collaborating with other NGOs, because sometimes a lot of these NGOs work in their little silos. They don't necessarily communicate, but there's strength in numbers. So maybe if they collaborated a little bit more, they might have some more power. Again, engaging with technology and development and also scientific guidance. So talking with us about what the priorities are, what we need to know, what kind of data we need so that we can feed into policy. Okay, so sticking on the theme of mapping marine litter, but kind of changing the location somewhat. This is a project, again, that I worked on during my postdocs. This was a project funded by the National Geographic Society called the Sea to Source expedition of the River Ganges, to map the abundance and distribution of plastic pollution in the Ganges. So the Ganges has been highlighted or identified as one of the key contributors, the key inputs of plastic debris into the global ocean. So we kind of wanted to understand how plastic within the catchment is making its way into the Bay of Bengal and then further out into the ocean. So we basically made our way up the Ganges from the Bay of Bengal in Bangladesh here. So we lived on a riverboat for a couple of weeks, and then we crossed the borders to India, and then we made our way by land, not walking, but trains and cars. We sampled 9 different sites in total, all the way up to Rishikesh in the Himalayas, which is a really cool expedition. So it was a really large project. There were researchers from the US, the UK, India and Bangladesh, and there were 3 kind of multidisciplinary teams. So you had the land team who were going out into the streets and kind of mapping the amount of plastic pollution on the streets, going into shops and kind of auditing how much plastic was in there, plastic packaging, how plastic was used within the community. We had a social team who was going out and interviewing people within the local communities, having focus groups, trying to understand how people use plastic and what their attitudes were towards it. And then I was in the water team, and our kind of key activities were mapping the riverbank for the amount of derelict fishing gear on the riverbanks, and also boat-based sampling of the water and sediment for microplastics. So I'm just going to talk about this one because I led on this one. So the aim was to examine the inputs, socioeconomic drivers and ecological impacts of derelict fishing gear in the river system. So after mapping, surveying the riverbank sites at the 9 different sampling locations, we found that the main kind of sources of plastic from fishing waste were in the form of strings, nets, ropes and floats, and these were mainly kind of polystyrene foam. And the most common type of gear discarded on the riverbank was nets by volume. And this is really important because that poses a significant entanglement threat to aquatic wildlife. And as before, I kind of mapped where the highest densities of this type of litter were. So the lighter the colour, the highest density. So you can see these 3 sites in Bangladesh had the highest densities of fishing related debris. And there's a couple of reasons behind this. Firstly, probably because this is a really important area for fisheries, That's the main source of income in that area. So it's very tidal, a lot of water movement between the Bay of Bengal and up in these lower areas of the Ganges. Very heavily fished areas. So obviously, the more fishing activity you've got, the more debris you've got. But also, probably relating to the socioeconomic factors of the region, it seems like India has a kind of stronger legislation and infrastructure, waste infrastructure, than this area of Bangladesh in terms of managing plastic waste. And then we spoke to the fishers through the kind of social surveys about why there's so much, like drivers in terms of why there's so much plastic waste on the riverbanks. And one of the key reasons is that they have a high gear turnover rate. So they either have to change their gear, replace their gear either 1 to 6 months or every 6 months to a year. So after a year, they basically have to just discard all of their fishing nets and get new ones, because they've used kind of materials that are cheaply imported and they break easily. So they have to just, they get to a point where they can't repair it anymore and they just have to discard it and start again with a new net. And when we asked them what they do with their old gear, around a quarter said that they just discard it into the environment. And these rates, these kind of responses were higher in these lower regions of the Ganges, so around Bangladesh. So yeah, they've got high turnover rates of the gear and then they just discard it into the environment. Other kind of outputs were they either burnt it or it got repurposed into things like fences. So then the next part of this study was to kind of do a biodiversity threat assessment and to understand which air-breathing vertebrates, aquatic vertebrates, were most at risk from the impacts and rates of entanglement of from fishing gear. And these were threatened freshwater turtles, the endangered Ganges river dolphin, and freshwater otters as well. And so from a kind of why is this information useful point of view, these are all animals of conservation concern, and highlighting that these are at risk from being entangled by plastic fishing waste is really important for kind of directing conservation efforts to where they're most needed. And in terms of identifying where all this fishing waste is accumulating, the sites in Bangladesh have actually been identified as potential candidates for a project where fishers are paid to bring their nets in and deposit them into a system where they can then be made into other things like carpet. And this project, this kind of initiative, has been shown to be really successful in places like the Philippines. So it'd be really cool if we can get that off the ground and actually remove those nets from the environment and not just have them going into the river. Okay, so there's no natural segue to go on to this next bit about plastic ingestion, so I'm just going to dive straight in and talk about plastic ingestion by marine mammals. So there's loads of different reasons why plastic might be ingested by marine vertebrates. So you've got things like active selection, which is driven by things like olfactory cues and visual cues, and olfactory cues are just things like smell. So when plastic's been in the marine environment for a long time, it can become biofouled, which then gives off a scent, and it means that it smells like food to a lot of different animals, and they can actually be driven to eat it because they're mistaking it for food. And the same with visual cues. It's that classic example of a turtle mistaking a plastic bag for a jellyfish. Then you've also got things like accidental ingestion. So that can be if plastic debris is mixed up in the water with normal food sources. So for example, with a turtle, you could have fibrous bits of plastic that are mixed in with seagrass, and there's a green turtle's kind of munching along through the seagrass and accidentally just eating the plastic as it's going. And then you've got indirect ingestion as well, and that's through things like trophic transfer and intergenerational transfer. And I'll talk about those a little bit in a minute. So in terms of the impacts, we know that macroplastic ingestion, so these are large bits of plastic debris that are larger than 5 millimeters in size. They can cause injuries to the digestive tract, like ulcerations, perforations, and lesions. And this is normally through things like hard and sharp bits of plastic that can kind of puncture the digestive tract. And then you've got obstructions from large volumes of compacted debris, and this is normally through things like soft sheet plastic that can be squished into a bit of a ball in the digestive system. This can lead to a false feeling of satiation, so the animal feels like it's full of food, but it's actually full of plastic, doesn't eat properly, becomes malnourished, and that can mean that they're more susceptible to other stressors like diseases. And then plastic that's in the environment can absorb chemical contaminants from seawater. There's also got contaminants added during production as well. And these can all leach out if they're ingested into biological tissue. And these types of chemicals have been shown to cause impacts on reproduction and immune system function. I think the next slide is a bit gory, so if you're a bit sensitive to things, I'll maybe look away. Yeah, so these are just, so we think that marine mammals, certain types of marine mammals may be susceptible to plastic ingestion more than others. So we think that deep diving species like sperm whales and beaked whales seem to eat more plastic and become more compromised by it. So these two examples are here of Cuvier's beaked whales that were found to have lots of plastic in their digestive system that actually led to their death. And this is the same with sperm whales. So there's been quite a lot of stranded sperm whales in the Mediterranean, and this is a juvenile sperm whale that washed up in Greece. And obviously you can see all the stomach, it's emaciated, and you can see the stomach is just absolutely full of plastic bags, burlap sacks, fishing ropes, and all sorts. And so these animals do seem to be more susceptible to ingesting macro debris. And then in terms of microplastics, so as I mentioned, these are plastic items that are less than 5 millimeters in size, and they can be ingested via two main pathways, either direct ingestion or indirect ingestion. Direct ingestion normally occurs for things like more generalist feeders, like the filter feeders, baleen whales and things that are engulfing large volumes of water and potentially also engulfing microplastics that are in the seawater. Indirect ingestion is a pathway that normally occurs more for predatory animals like the dolphins, the seals. You could also have predatory fish like tuna and the sharks and things. So this is where microplastics that are either in the seawater or the zooplankton are eaten by secondary consumers like fish. And then these fish are eaten by the top predators, and then the microplastics are essentially passed up the marine food web. This might seem fairly obvious, but until I did my PhD, this had only really been demonstrated in low trophic level animals like crabs and mussels. So I wanted to look at it in terms of top predators. So I analysed the poos, scats from seals from the Cornish Seal Sanctuary and the wild caught fish that they were fed on for microplastics. And this is kind of the scenario of looking at captive seals is the closest you're going to get to an enclosed kind of system, like a lab experiment, but obviously it's large marine vertebrates, so you can't get that. But these animals weren't exposed to plastic pollution like wild seals might be. So the presence of any microplastics in their scats was kind of an indication of this trophic transfer mechanism occurring. And I did find microplastics in both the fish and the seal scats, so we kind of identified this pathway of exposure. It's worth just pointing out as well that these animals, these filter feeding animals, are probably exposed to both direct and indirect ingestion of microplastics, because if they're also ingesting zooplankton that contain microplastic and seawater that's contained microplastic, then they're getting it from both pathways. So after kind of identifying that these captive animals in the seal sanctuary were being exposed to plastic pollution, microplastic ingestion, I wanted to look at wild marine mammals. And so to do this, I worked with the stranding schemes from around the UK, the Scottish Marine Animal Stranding Scheme and the Marine Strandings Network and the Cetacean Stranding Investigation Programme, to look at the digestive tracts of different species that stranded around the UK coast. So lots of different marine mammal, dolphin and seal species, but you can see the most common kind of species that I looked at were common dolphins, which are the red dots. And this is kind of indicating where they stranded, and harbour porpoise as well, which are the green dots. And they're kind of spread around the UK. So when an animal stranded, this is a harbour porpoise, a post-mortem was carried out, and the cause of death was identified if it could be. The digestive tract was extracted and given to me, which I then processed in Plymouth Marine Lab inside this kind of clean lab setup to avoid any kind of airborne contamination. And to cut a long story short, I then used enzymatic digestion to remove some of the organic material, looked at it under a microscope, picked out anything that looked vaguely like a piece of plastic. These are microscopic, by the way, and used a technique called Fourier transform infrared spectroscopy or FTIR for short, to identify whether the particles that I picked up were actually plastic or synthetic or natural, and if they were synthetic, what kind of polymer type they were actually made up of, because that kind of information helps us to identify potentially where they've come from. And if all goes well, you end up with little particles that look like this under the microscope. And yeah, as I said, I use FTIR to confirm that they are actually plastic. It's amazing that natural things like sand can actually look very artificial when you look at them under the microscope. So I did start to lose my mind a little bit when I was looking at them. So it's good to have that confirmation. So in terms of what I found, so I found microplastics in every animal that I looked at, with an average of around 5.5 particles per animal and a maximum of 12. So this plot just shows the number of microplastics along the bottom and the frequency at the side. This was actually quite a lot lower than we were expecting. I think, you know, we all think about microplastics as being everywhere within the marine environment. But I think, yeah, it is a bit lower than what we were expecting, but this was 5 years ago, 6 years ago, probably when I was doing this, and the methodologies for looking for microplastics has really come on since then. And our ability to look for smaller size classes has really developed. So I think if we were looking for smaller microplastics, we'd probably find more of them. I only found macroplastics in one of the animals I looked at. So this was just some green netting in the stomach of a common dolphin. So to go back to those kind of low numbers, I also did look at kind of where the microplastics were in terms of the digestive tract section. So this is just the section along the bottom and the number of microplastics. And the stomachs did have slightly higher number of microplastics, potentially indicating it's a retention site. But they are obviously eventually moving through the system, we think. The other explanation is that microplastics could be translocating out of the gut and potentially making their way into other organs, which I'll come on to a bit more in a minute. And then as we had information on cause of death because of the post-mortem, I couldn't not look at this in relation to the number of microplastics in these animals. And we found that animals that died of infectious diseases had a slightly higher number of microplastics than those that died of trauma. So by trauma, it's things like bottlenose dolphin attacks or other kind of, traumatic, like bycatch, for example. But I always say to take this kind of result as a pinch of salt. We don't really know what the ecological and the biological consequences of this it is. I mean, it's quite a small sample size. It's a very messy sort of system to analyse. It could be that the animals that died of infectious diseases just weren't eating normally at their time of death, and so could be more likely to ingest plastic. So a lot more work is needed to kind of understand the sublethal effects of microplastic ingestion on these animals. And so, yeah, as I said, I used FTIR to look at the polymer types. I only did it on a sample, subsample of these, but we found that most 61% of the particles were nylon and 10% were PET and polyester. And these are polymers that are commonly used for everyday items like fishing ropes, our clothing. The PET is used for food and drink packaging. So this kind of gives us an indication of where these microplastics might be coming from. And then you might notice that some of the animals that were on my species list were quite small dolphins and seals, and we're obviously missing a huge demographic of animals like the large filter feeding animals, the deep diving whales. There is a good reason for this, and it's namely their size. So this is, as I said, this is the harbour porpoise intestines, and this is the intestines of a sperm whale that stranded in Wales, I think it was last year, which is hundreds of meters long. And it's literally like looking for a needle in a haystack. Obviously, we can't sample. I did the whole digestive tract of this animal. We can't do that with these huge animals. We just don't have the facilities to do it. You need quite a clean environment to be able to avoid contamination. And you don't always have the luxury of being able to spread the intestines across the beach to actually get access to them. So we want to know, like, how do we even go about sampling these huge animals that in a representative way that we can then extrapolate up and identify kind of how much microplastics they're exposed to. So this is me and a previous master's student trying to subsample the intestines of a juvenile minke whale. We decided to kind of section up the intestines by 20 centimetre sections and then sample each every other one, so try and do the whole one. But this is actually quite a small animal. This was in an abattoir, so it's not completely clean, but it's cleanish, and we can at least kind of sample the air to understand how much microplastics is exposed to our samples. But this is the fin whale that stranded was it in November on Newquay Beach. And this is my wonderful PhD student Emma trying to dig around in the intestines, trying to kind of work out how to get access to which bits to even sample. And in this case, the tide was coming in. There was lots of people watching. We didn't have the luxury of pulling the entire length of the intestines out and trying to work out what the overall length was so that we could even understand how much we were sampling. So she's in the process. She's got the very glamorous task of trying to understand how we can robustly sample these animals that's representative, but also practical for the stranding scientists who are working with us and providing these samples. So yeah, try and kind of understand the exposure levels of microplastics to these animals. So on kind of to close on this, I just wanted to kind of run through things that people are always wondering, like, so what? Like, these animals are ingesting things like indigestible items all the time, like squid beaks and fish bones. Are like tiny pieces of plastic really going to have that much impact on their health? Obviously, I don't know the answer to that, but it's something that we're trying to work on finding out. But in my view, there are kind of three main aspects that we need to look into. So the first is mechanical, sort of like the physical presence of microplastics. What are they actually doing? So until recently, I always sort of said they might be passing through the digestive tract, or very small microplastics might be able to translocate through the gut wall and make their way to various organs. And recently, a study from a colleague at Duke Marine Lab has found microplastics in the blubber, the lung, the acoustic fat pad, and the melon of marine mammals. So they are somehow getting out of the gut and into these other tissues within the animals' bodies. And obviously, we don't know at this point what kind of impact it's having on the functioning of those organs, and we need to look at other organs as well to understand their impact. We do know that microplastics and nanoplastics, so these are really, really tiny microplastics, are known to disrupt the gut microbiome and the gut barrier function in things like mice and fish species that have been studied in the lab. Obviously, we can't do that with marine mammals, but it's kind of a first step to understanding that. As I mentioned earlier, plastic in the environment has chemicals on and within it that can desorb into biological tissue when it's ingested. And I want to know kind of to what extent does plastic ingestion expose animals to chemicals aside from their normal kind of dietary and environmental inputs? It's not as easy as just kind of counting the number of microplastics and then saying, you know, this X number of microplastics equal Y amount of toxin in the tissue. There's lots of different factors that can influence that relationship, like the polymer type, the age of the plastic, obviously the different types of contaminants that are already in the area. There's a time lag between the plastics being ingested from the animal and the tissues showing up with a chemical signal. So it's a really complicated relationship, but I'm hoping one day we can look at that. And then lastly, plastic in the environment be colonized by a whole range of different types of bacteria. And we don't know whether the ingestion of plastic can expose animals to various different pathogens and diseases. So one study that looking at coral reefs found that the vulnerability to disease increased from 4 to 89% when the corals were in contact with plastic pollution. So plastic ingestion could act as a potential vector for diseases as well. So yeah, we're only just kind of scratching the surface in terms of what the impacts of plastic pollution are on marine mammals. So I just wanted to highlight some of the future work that we're doing. So as I mentioned, Emma is currently starting her PhD looking at the extent of plastic ingestion by ocean giants, so not just marine mammals, but other kind of marine megafauna like sharks and large turtles as well, and kind of trying to understand how we go about sampling them. And then circling back to the kind of more marine mammal conservation and ecology side of things, without the plastic, Helen Chadwick is looking at marine mammal bycatch in the southwest England. And so her research is working off data that's been highlighted from the stranding schemes that the southwest seems to be a hotspot for bycatch of small cetaceans like common dolphins and harbour porpoises. she's created these maps, and she's just at the start of her PhD as well. And she's going to take an interdisciplinary approach to kind of understanding what the drivers of this are. So she's going to be using marine mammal ecology and conservation. She's going to be working with the stranding schemes again to look at veterinary pathology data, potentially also looking at how bycatch incidences affect the welfare of the animals. Obviously, it's not good, but at what kind of scales. She's also currently up at Heriot-Watt University in Orkney, to work with oceanographers up there to try and model where the interactions between these animals and the fishing and the fishery might be occurring. If we're identifying carcasses on the beach, where might have those come from, so we can identify the overlap of those kind of interactions. And then hopefully that's all going to feed into some policy and social science to kind of work with the fishers and understanding what mitigation measures might be viable options for them, and kind of work out the cost benefits as well to try and tackle this issue. So just to finish off, I wanted to thank all my collaborators. Obviously, this is a lot of work that's been had a lot of input from different people and different organisations, different funders, and just come back to my original slide. I still don't know who I am. I sort of dabble in ecology, conservation science, hydrology, social science, geography, and marine biology. But yeah, I'd say I'm a jack of all trades and master of none. So thank you very much for listening.
Thanks so much, Sarah. That was great. Do we have a question?
That was great, thanks. I was just wondering, in strandings, do they take samples for chemical analysis, so to look for bioaccumulation of all sorts of things? And is there any signal coming out of that might point towards the role for plastics?
They do. And it's kind of archived at the moment. It's something that I wanted to look at through my PhD, and we looked into it. It's very expensive, sort of prohibitively expensive. I wouldn't have been able to afford to do it during my PhD. So they do have the samples, and obviously we have the kind of microplastic abundances from the digestive tracts information, and we just need to have the analysis to marry those two things up, or at least even try. Some people have tried it with seabirds and have detected a slight signal. Other people have done it and not detected anything. So it's a bit of a dark art at the moment. It's very new and not well established. So I have just applied for funding to try and do this. And I think Emma's going to probably try and scope out ways to do it at later stages of her PhD as well. But it is the kind of one of the main questions that's kind of hanging over us, but it's just so expensive and so like risky in a way, because there's so many other, even just contamination of chemicals during the lab processing.
Yeah.
Exactly. There's plastic everywhere.
Yeah.
So you have to be really stringent, and we really just need to get to a point where we can even just develop the methodology and then try and roll it out to actually some kind of assessment as well. But that is in the pipe if I can get some money to do it. I'm trying. Yeah. Thanks for your question.
Sorry about
That's fine.
Sarah, I'm just wondering, like, why the focus on microplastics especially? Is this because a lot of these species actually don't have microplastics, or somebody else is doing it, or I was just wondering.
I think with the macroplastic side of things, we know that they're definitely having an impact. It's very black and white. Some animals are quite obviously compromised by eating macroplastics, but then there are areas like around the UK coast and certain foraging behaviours where animals just aren't ingesting macroplastics. But we know that microplastics are literally everywhere. And so I guess the kind of questions around microplastics are about everyone's assuming that they can cause harm, but we don't really know what that is or if that is in these animals. We do know that in low trophic level animals, it does have impacts and significant impacts, but for these animals, we just don't know. So I think from a curiosity point of view, it's like, I want to find out, but also if we're going to invest lots of energy and resources into highlighting this as an issue, then really we do need to understand the real world impacts rather than just kind of assuming that it's causing harm.
I'm going to let you stand there so that people online can see you. Sorry.
It's whether you think it's like all the microplastics and nanoplastics inside the body come from survive compared to the lungs?
Yeah, I mean, that's another question as well. I want to one day kind of look at is what, obviously we know that microplastics are floating around in the air, we're breathing them in right now. It's all of these things in this room. So obviously we are potentially inhaling quite a lot of plastic, and I do wonder whether some of the microplastics that study that I mentioned about in marine mammal lungs, they might have ingested, it could have been from contamination as well, because they weren't collected specifically for microplastics research. You have to be extremely stringent about contamination control. So there are some doubts around that, and that's why we need to keep doing more kind of robust research on it. Yeah, I would quite like to do some at-sea sampling of air and seeing kind of what the exchange of microplastics into water and into the like air-breathing marine vertebrates are as well as a potential pathway. But again, that seems very kind of nebulous at the moment. So trying to go for the low-hanging fruit.
Darling, do you have?
Yeah, it's striking how much the nylon was the biggest thing. But in terms of contamination, presumably on land, there isn't really. Isn't that just the fishing?
No, it's
So it could be
in
used
our
for-
clothes.
not-
Yeah, it's everywhere. And I mean, we looked at that kind of polymer composition. It does match up quite well with other studies, but then there's so much diversity in just the environment and the production of plastic.
You can't.
You never have exact markers. We did also look at things like colour and size, and blue and black were the most common colours, and that really strongly overlaps with other studies looking at just the general marine environment, but also ingestion by things like fish as well. And that could be because those colours are very common in production of plastic, like our clothing, but it could also be that actively being selected by fish and other organisms to eat because they look more natural. Don't know. But yeah, so it's kind of, it's nice to know that those things at least overlap, but with the polymer types, it's quite often among the studies, it's always pretty different, just because there's so much diversity. Yeah. Yeah. Questions?
I'm curious. It's a more a natural history thing. I was just interested by your deep diving species, right? And it just struck me that, you know, what do you think the mechanism might be there? Do you think they might be just be using shading, low light levels to pick up prey items?
Yeah, so these animals use acoustic information through sonar echolocation to identify their prey. They're normally hunting things like squid in the dark, in the depths. So we've been having lots of discussion about this, and part of me wonders whether the type of plastic they're ingesting is a lot of kind of sheet plastic, which may be less likely to float at the sea surface because it can become biofouling. It hasn't got those kind of air pockets like a bottle would do, for example, to keep it at the surface. So then it's coming down through the water column and obviously overlapping in terms of the distribution where those animals are hunting. Also, I guess a piece of sheet plastic kind of moving down through the water might look from an acoustic signal like a squid.
Quite a big signal.
Yeah, exactly.
Yeah.
And so, yeah, Emma's working on a systematic review at the moment, looking at kind of foraging behaviours and foraging techniques to try and identify which species are, you know, what those ecological drivers are of ingestion. And we're trying to kind of pinpoint that exactly. But I don't think we'll ever know. But I think that's my theory, is it's kind of just the confusion thing.
Yeah, please.
Thanks for a really interesting talk, Sarah. So I was just wondering, with the feature scheme data, I wonder if you picked up kind of any seasonal trends in terms of quantity of plastics on beaches.
I think casting my mind back, I think the autumn had high levels of marine debris on it. So it's a bit funny because the data collection was mainly in the September time, because that's when MCS have their Beach Watch event. I think it's the 3rd or 2nd weekend of September. So you do have peak volunteer activity at that time anyway. But obviously, the analysis that I did hopefully took some of that kind of bias out of the data. But yeah, it seemed like kind of autumn period did have higher levels, and that's probably because of the storms. But it kind of depended on the locations as well. So obviously the Atlantic facing or more exposed kind of beaches had higher levels during the autumn, and potentially more sheltered beaches had higher levels in the summer because of more people on beaches. Yeah, it's a very messy data set, very insightful, but also quite messy.
Questions?
I'm just sort of thinking about the timescales. Do you have any sense? I don't know whether you can get any kind of temporal element to the land doing this. So if you're doing these beach cleans, are we picking up on things that have been floating in the ocean for 20 years?
Yeah.
You know, these, we think, I guess that may differ as well. If these are exposures to fishing in the
Yeah.
Atlantic, might be quite different to
Yeah.
North Sea wet-like issues.
It's not something that I was able to look at with this data set because the beach clean citizen science data is very kind of zoomed out view. It's literally just abundance, and they were able to, you know, the volunteers were able to categorize the litter types. But when we were discussing it with the organisations during the workshop, we were sort of talking about whether it would be possible to, if you had some very keen volunteers who wanted to do more in-depth data collection, could they collect information on stuff relating to the date of the items, because like things like crisp packets have got best before dates on, and you can identify whether it's literally just been dropped on the beach just by looking at it, or whether it's been in the ocean for a long time and then it's washed up. So that is something that we've been talking about, but not been able to achieve with this data set. But yeah, it would be a cool thing to do, because differentiating between whether it's come from the sea or just being dropped on land has policy implications as well.
Do you see a difference in the plastic burden in animals in the UK versus, because you said it was quite low, like compared to like, say, animals from the Mediterranean basin where it's like really closed kind of place that gets a lot of like plastic pollution?
Yeah, I've never done a direct like data comparison, but anecdotally, it does seem like we're in a relatively low area of concentrated microplastics compared to other, like we know that the Mediterranean is a hotspot for plastic debris. It's an enclosed sea, it's surrounded by very high population centres as well. Yeah, we know it's a hotspot, so it's likely that animals that are in there are being subjected to a lot more of plastic exposure than our animals are around the UK coast. Obviously, some of our animals migrate like around as well, so it'd be interesting to know where they encounter hotspots. But I'm working with the International Whaling Commission to try and kind of identify hotspots of marine litter, plastic pollution, and cetaceans to see where the overlaps might be in terms of identifying vulnerable species and populations for that purpose.
Thank you so much, Sarah. That was great.
You're welcome.
Thank you all for coming as well. Thank you online.